Hello everyone, hope you guys match your anatomy today. Regarding your BCH, I spoke to your lecturers regarding you guys area of concentration, and then your lecturer told me that questions are literally going to emerge out from your slides. Okay, so all you're just going to do, study your slides effectively, and then maybe you literally upload it in any AI, telling it to generate all possible questions they will likely ask regarding the content in the slide. So I just felt that there are some things I literally need to just touch in biomolecules, like maybe disorders and stuff like that, and some things that I didn't have time to teach you guys physically. Um okay, so on introduction, we can still remember our biomolecules, stuff like that, their introduction and metabolism. Okay, so I just think that I need to give you guys a summary table regarding some disorder associated with some kind of biomolecule metabolism. So firstly, this is a summary table for protein and amino acid metabolic disorders. Okay, so this is very simple. Okay, so just know the disorders, the enzyme deficiency and pathway affected. Okay, I feel with that you're good to smash your BCH or MCQ. So for instance, now we have this, which is albinism, a disorder, and then the enzyme deficiency is the thyroxinase, and then pathway affected is melanin synthesis. Then we have two, that's phenylketonuria, and then the enzyme deficiency is phenylalanine hydroxylase, and then pathway affected is phenylalanine conversion to thyroxine. Then we also have thirdly homocystinuria, and then enzyme deficiency is cystathionine B synthase, and then the pathway affected is the methionine pathway. Then we'll have the maple syrup urine disease. Okay, the enzyme deficiency is the branch chain amino acid dehydrogenase complex, and we remember the branch chain amino acid like the valine, leucine and isoleucine. And then the pathway affected is the branch chain amino acid metabolism. Okay, so we also have alkaptonuria, when the urine look dark in color, stuff like that. And then the enzyme deficiency is the homogentisate 1,2 dioxygenase. Then we have the pathway affected is the thyroxine and phenylalanine degradation. Then we also have tyrosinemia type 1, and then the enzyme deficiency is the fumarate acetoacetate, and then pathway affected is the tyrosine catabolism. So we have cystinuria, and then the enzyme deficiency is the SLC3A1 transporter, and then the pathway affected is the renal amino acid transport. Then we have ornithine transcarbamylase from the urea cycle stuff, and then enzyme deficiency is the ornithine transcarbamylase. Now the pathway affected is the urea cycle, already know that. And then we go to the heart knob disease. Okay, the enzyme deficiency is the SLC 619 transporter. Okay, neutral amino acid in the intestine, stuff like that. So let's move on. The last time I taught you guys intro to nucleic acids. Okay, and then there's something I asked you guys to take note of the other time: donors of atoms in purine and pyrimidine, as well as nitrogenous base in their nucleoside and nucleotide form. Okay, so let me just do a quick recap. I told you guys that we have two nitrogenous bases, the purine and pyrimidine, right? And then we have two purine nitrogenous bases, which is the adenine and guanine, right? And then the pyrimidine, the cytosine, thymine and uracil. Okay, we already talked about the one in DNA and the one in RNA. Okay, we have adenine, guanine in both DNA and RNA. We have cytosine in both DNA and RNA. But the difference is that we have thymine in DNA, and then we have uracil in RNA only. Okay, so the nucleosides, difference between nucleoside and nucleotide. Okay, we know both of them have nitrogenous bases, they both have pentose sugar, but the difference is that the nucleoside has no phosphate group, while the nucleotide has a phosphate group. Okay, so we'll just talk about in each of all these nitrogenous bases, their name in nucleoside form and then in nucleotide form. Okay, for instance, the adenine in its nucleoside form is called adenosine, while the nucleotide form of adenine is adenylate, or you can also call it adenosine monophosphate, diphosphate, or triphosphate, depending on the number of phosphate group in A. The same thing happens to guanine in its nucleoside form is guanosine, and then in its nucleotide form is guanylate. You can also call it guanosine monophosphate, diphosphate, triphosphate, depending on the phosphate. Same thing applies to the cytosine, thymine, and uracil. When you see them in their nucleoside form, we call it cytidine, thymidine, and uridine. While in their nucleotide form, you call it cytidylate, thymidylate, uridylate, or you call them cytidine monophosphate, cytidine diphosphate, triphosphate, depending on the number of phosphates. Same thing, thymidine monophosphate, diphosphate, triphosphate, uridine monophosphate, uridine diphosphate, uridine triphosphate, depending on the number of phosphate group in it. So now the bonds in nucleotide. Okay, so bond in nucleotide. See something. So the bond between nitrogenous base and pentose sugar is called glycosidic bond. Okay, the bond between nitrogenous base and pentose sugar is called glycosidic bond. The bond between purine nitrogenous base and the pentose sugar is A9 to C1, which means that's the ninth nitrogen and carbon 1 of the pentose sugar. Okay, while that of pyrimidine is between the first nitrogen and the carbon 1 of the pentose sugar. Okay, take note of this glycosidic bond between the nitrogenous base and pentose sugar. So now the bond between pentose sugar and phosphate group is called the phosphoester bond. Recall the one between the nitrogenous base and pentose sugar is called the glycosidic bond. If it's for purine, is N9 to C1. If it's for pyrimidine, is N1 to C1. So let's move on. Donors of atoms in purine and pyrimidine rings. Okay, you can see clearly the formic acid donate the second carbon and the 8th carbon in the purine ring, while glycine donates the 4th and 5th carbon as well as the 7th nitrogen in the purine ring. CO2, which is carbon dioxide, donates the 6th carbon, and then aspartate donates the 1st nitrogen, and then glutamine donates the 3rd nitrogen and the 9th nitrogen. Okay, why if we go to pyrimidine rings, the second carbon is donated by CO2, the 3rd nitrogen donated by an amino group, and then the 4th, 5th, and 6th carbon is donated by aspartate. Okay, so you can literally pause and do a revision on clinical correlate associated with fatty acid. I told you guys about the Revsum disease and phytanic acid the other time, as well as Wilgars. Okay, I told you the Wilgars is as a result of deficiency of peroxisome. Okay, which is an organelle that play a role in very long chain fatty acid oxidation. Okay, and then I told you guys about phytanic acid being a branch chain fatty acid with 20 carbon. And then the last time in class I told you that Revsum disease occur as a result of deficiency of fatinoic A hydroxylase, which is an enzyme that degrades phytanic acid. When that enzyme is deficient, it leads to buildup of phytanic acid. Okay, which we call Revsum disease. So in such case, the person tries to avoid food or diets rich in phytanic acid, like meat from ruminant animals or fat from ruminant animals should be avoided to reduce intake of phytanic acid. Okay, you can just pause and do revision on these. So the DNA, this is the structure of the DNA. I remember the last time I told you that the DNA has double helix and is double stranded. Okay, so take note of something. There's something we call complementary base pairing, right? So what it means is that two nitrogenous bases that are compatible tends to pair with each other. Okay, now I use a mnemonic in remembering this. Okay, you see the A and T. A is adenine and T is thymine. So A and C, which is adenine and thymine, are complementary base. So two of them likely pair with each other, okay? So how do I remember this? I use apple in a tree. You will see apple in a tree. A for apple, which is adenine, and then T for thymine, which is tree, okay? Two of them are complementary base, so they pair. And then C and G, which is cytosine and guanine, are also complementary base pair. I use car in a garage, right? C is car, while G is garage. Okay, so apple in a tree, then car in a garage. So adenine and thymine pair, and then there's a bond between the two of them. Okay, the bond is hydrogen bond. I'll have two hydrogen bond between adenine and thymine, and then we have three hydrogen bond between guanine and cytosine. Okay, take note of the complementary base pair and then the bond between them. Okay, between adenine and thymine, which is the two complementary bases in DNA, we have two hydrogen bond. Between cytosine and guanine, we have three hydrogen bond. Okay, now the bond between phosphate group, so we have high energy bond, stuff like that, and bond within this helix is the phosphodiester bond. Okay, which run 5, you must have been heard about 5 prime to 3 prime, and then 3 prime to 5 prime. So how it runs, it runs like anti-parallel. Okay, the backbone runs, this sugar phosphate backbone run anti-parallel. Okay, from 5 prime to 3 prime end, and from the other aspect is 3 prime to 5 prime end. Okay, they run anti-parallel. Okay, so let's move on. So you can literally pause this to just do a recap on disorders of purine metabolism, as well as disorders associated with pyrimidine metabolism. I taught this the last time in class, so you can just pause and take note of this. Okay, so the last thing I want to talk about is gluconeogenesis. Okay, this is something you need to know. Questions are likely asked from gluconeogenesis is that they might literally say, okay, what are the three, how many reactions are being bypassed, stuff like that. Okay, and then you need to know the reaction that are being bypassed. The reaction that are bypassed are literally those reaction in glycolysis that are irreversible. Okay, we know how gluconeogenesis happen, right? And then we know that gluconeogenesis is literally production of glucose outside carbohydrate precursor, and then we know that there are some irreversible reaction in glycolysis that are being bypassed. Okay, so we remember pyruvate go into the mitochondrial and get carboxylated to oxaloacetate by the enzyme pyruvate carboxylase, and then this oxaloacetate get acted upon by phosphoenol pyruvate carboxykinase to form pyruvate, and then the other reversible reactions in glycolysis get reversed. When we get to the point, this other point again, fructose 1,6 bisphosphate. Okay, from fructose 6 phosphate to fructose 1,6 bisphosphate is irreversible, so we literally still bypass this by an enzyme called fructose 1,6 bisphosphatase. Okay, fructose 1,6 what bisphosphatase. So it converts fructose 1,6 bisphosphate back into fructose 6 phosphate. I'm sure this is literally the regulatory enzyme, more of like the key enzyme, the rate limiting enzyme in the whole reaction. Okay, and then the last irreversible step is from glucose 6 phosphate back to glucose. Okay, and then we have an enzyme that converts glucose 6 phosphate back to glucose. We call it glucose 6 phosphatase, right? And this enzyme is present in the smooth endoplasmic reticulum of the liver. So this enzyme glucose 6 phosphatase is absent in muscle, so which is why muscle can undergo gluconeogenesis. Say something, and then when this enzyme is deficient, when glucose 6 phosphatase is deficient, the person suffer a condition we call the von Gierke disease. So von Gierke disease as a result of deficiency of glucose 6 phosphatase. So what happens is that there will be a build-up of glucose 6 phosphate, which means this guy can be funneled or channeled into other pathways like HMP shunts and stuff like that. So if they ask you how many reactions are being bypassed during gluconeogenesis, just know it's 3 reaction. Okay, it's 3 reaction. So you can literally just pause and do an overview over it. Okay, So I wish you guys good luck. Remember, in MCQ it work with speed and what accuracy, so smash your BC reach. Okay, I wish you good luck. I wish you guys good luck.